Assalamu alaikum. Inshallah, today I will introduce you to a new strain of Klebsiella pneumoniae. This strain is called hypervirulent Klebsiella pneumoniae. As all of you know, WHO declared Klebsiella pneumoniae as a clinical priority microorganism. It's not only causing pneumonia, but other infections too, like meningitis, bacteremia, and it's always associated with high morbidity and mortality. So our first research was trying to find out what's the percentage of ESBL producing Klebsiella pneumoniae in Asyut University Hospital. And it was really, we took like 130 samples from different nosocomial infections. And the rate was really very high, like a third of the cases were due to ESBL, multidrug resistance Klebsiella pneumoniae. Well, do we have some endemic clones in the hospital? This was our second question. So we did a research about the epidemiological typing of the MDR Klebsiella, and we took the isolates from pediatric VEP patients. As you can see, we took 20 clinical isolates, and we took also 4 environmental isolates from beds and commodes and floors. And then, although there were multiple clones, but we could find that there has been certain clones that were common in patients and environmental samples. 4 clones were characterized for group A, and 3 strains were of the group C. So when we took a closer look at those isolates comprising the group C, the 2 of them were environmental isolate from bed and the commode, and the third was from a patient. They have been, the isolates were taken in a sequential manner. When we take a closer look at their antimicrobial resistance phenotype, we could see accumulation of resistance pattern. So the first isolate was from a bed, and it was only resistance to penicillin. The second one was from a commode, and it was resistant to penicillin, cephalosporins, carbapenems. And the third one was from a patient. It was resistance to nearly all the available antibiotic. The second group A, one was from an environmental isolate, the 3 was from clinical patients, and again there has been accumulation of resistance. This posed the questions, how they accumulated the resistance? If they are the same isolates, why did they accumulate resistance? So we have 2 mechanisms of resistance. First is the chromosomal mutation, and the second is the acquisition of resistant plasmid. What is a resistant plasmid? It's an extra chromosomal DNA that has the ability to capture resistant genes and accumulate resistance. Also, this resistant plasmid can move from resistant bacteria to susceptible bacteria through a process called conjugation. And this conjugation leads to dissemination of resistance. So, do Klebsiella pneumoniae have this transferable resistant plasmid that's responsible for the accumulation? This, where we did our second research on the plasmid-mediated fluoroquinolone resistance in Klebsiella pneumoniae. And we found there has been high frequency of these plasmids in Klebsiella pneumoniae, like about 100% of the isolates harbor 1 or more of the plasmid-mediated fluoroquinolone resistance. Then we had another question. Do these plasmids have another resistant genes other than fluoroquinolone? Does these plasmids have carbapenem also? What we found that these plasmids have carbapenem and fluoroquinolone resistance in 90% of the Klebsiella pneumoniae isolates, and when we did conjugation experiment, they have been transmitted at a high rate. 100% of these genes have been transmitted with each conjugation experiment. So we fight not only Klebsiella pathogens, but we fight also multidrug resistant plasmids. So what you know is what we're talking about in the past years was known as the classical Klebsiella pneumoniae. This is the hospital-acquired Klebsiella. It is of low pathogenicity. It just causes only mild pneumonia, but it's resistance to antimicrobial drugs. The new strain that appears in the late 80s, it's called hypervirulent Klebsiella pneumoniae. It is of high pathogenicity, but susceptible as any of the community-acquired organisms. susceptible to drugs but cause severe infections. First, it causes community-acquired necrotizing hepatitis, necrotizing pneumonia. Unfortunately, it has been seen in the hospital now and causing severe necrotizing multi-drug resistance pneumonia. So how they develop? The classical Klebsiella has a plasmid that carries resistant genes, and the hypervirulent Klebsiella has plasmid that carries variant genes. What has happened that when these two pathogens, they meet in a single patient, the two plasmids combine, and we have this hybrid resistance variance plasmid that caused us to have a multi-drug hypervirulent Klebsiella pneumoniae that we see now. So our second question was, do we see these multi-drug hypervirulent Klebsiella in our patients, in our hospital? So we did this research looking for the hypervirulent Klebsiella in VEP patients isolated from chest ICU, and the results were like 66%, like 2/3 of the Klebsiella pneumoniae were multi-drug resistance and hypervirulent. So we have a predominant hypervirulent strains. Also, when we did PFGE typing, trying to find if there is a relationship between the clones, we found a certain class 6. 5 of the isolates have been taken from pediatric and the 6 from the chest ICU, which means that we have circulating pathogen in our hospital. And the 2 isolates have been isolated 1 year apart, like in the chest were like in 2017, in the pediatric 2018, which means that we have in our hospital an endemic hypervirulent Klebsiella pneumoniae. Also, another cluster 7, 2 of the isolates were classical and 3 were hypervirulent, which means that there is something here causing the evolution of the commonly known classic Klebsiella pneumoniae into hypervirulent Klebsiella pneumoniae. So do we have circulating hybrid plasmid that caused this evolution of the classical Klebsiella into hypervirulent Klebsiella? So this is where we went to whole genome sequencing. We took 4 isolates, 2 of them of the same type, and the others are different. We were looking for the different plasmids in these isolates. We were trying to find out only single circulating plasmid. But to our surprise, we find like 1 multi-drug resistant plasmid in 3 isolates, another multi-drug resistance in 3 isolates, and then we found what we were looking for, the hybrid virulence resistant plasmids in 3 isolates. This is how it looked. It has resistant genes and it has virulence genes, and it has been present in 3 isolates. This is what we call the hybrid plasmid that has been found in 3 of the 4, like 75% of the isolates that we looked for in it. Is this hybrid plasmid responsible for the evolution of the hypervirulent Klebsiella? Well, we had 2 isolates of the same sequence type, K17 and K30. They have been isolated from your chest ICU 6 months apart. The first one was classical, the second one was hypervirulent. So how did this happen? When we looked closer, we found that Klebsiella 22 was in the middle, isolated in the middle, and it has the hybrid plasmid, so it must transmit it through conjugation to the classical Klebsiella and converted it into the hypervirulent. The other multi-drug resistant plasmid was so funny that we found it in an Egyptian patient who lives in the United States, but he had paid a visit to Egypt in 2012. The other resistant plasmid has been found in Canadian patient who just has been hospitalized in Egypt just 2 days, which means that these circulating plasmids do not only present in our hospital, they cross the border of the hospital, they are present all over Egypt, they even cross the borders of the countries. So what we have reached that our previous community in the hospital was classical Klebsiella pneumoniae, which is mild Klebsiella, although resistant to antibiotics, but just cause mild pneumonia. But because of this hybrid plasmid that reached the hospital, we don't know when, we don't know how, but it's here and circulating. We have circulating multi-drug resistant hypervirulent Klebsiella that transformed most of the community into resistant hypervirulent, and this is why we see severe pneumonia cases that do not respond to traditional treatment. So, how to combat this? This is a huge thing. How to combat? The conventional antibiotic that people use, even if it's combined, dual antibiotic or triple, the bacteria resist them immediately because they overexpress what's called efflux pump, so they expel the drug outside and they become resistant. So we thought of another solution. It's outside the box. It's just only research, but we hope one day we can see this on the practical use. It's the loading of antibiotics on nanoparticles. And this is our second line of research. We loaded antibiotics into nanoparticles and exposed the bacteria to these nanoparticles. Our hypothesis was that the smaller size of the nano-sized antibiotics will go inside the bacteria and kill the bacteria before it has the ability to express the efflux pump. We had a number of research on this to test our hypothesis. First one, we tested the levofloxacin loaded nanoparticles on E. coli, and it greatly decreased emergence of resistance. The second research that we did combined antibiotic, meropenem and ciprofloxacin on E. coli, and we found that there is marked decrease in the efflux pump, multidrug resistance, and emergence of resistance. Our third work was on a E. coli septic rat model, and it improved the survival rate from 70% to 100% survival rate. Okay, do this thing, this is E. coli, so it's, some people could say it just is not highly pathogenic microorganism. What about the hypervirulence? Okay, we took this to the second step, and our ongoing research now is about the combined nano-cipro-meropenem on multidrug-resistant hypervirulence. We did in vitro testing called time-kill curve, is that we exposed the bacteria to the combined antibiotic, and then we measure the number of the bacteria against time. The first line, the blue line, is the free conventional antibiotics, as you can see, and this is, we tried the first on the susceptible Klebsiella that is susceptible to all the antibiotics. Although first, the blue line of the free antibiotics, there has been at the beginning killing, but as you can see, the bacteria did not never reach the baseline. On the contrary, for the nano-sized antibiotic, the pink line, there is extensive bactericidal effect and the bacteria reach the basal line. Why? Because the conventional antibiotics stimulate the spontaneous mutations and they will not be able to kill the mutants, even in a fully susceptible bacteria. When we tried the multidrug hypervirulent Klebsiella, it was the contrary. The conventional antibiotics, the blue line, has no effect on the bacteria. So if you have a patient in your ICU having hypervirulent Klebsiella and you give him 2 antibiotics, you're just giving him candy. Nothing has happened. But when we looked at the nano-sized antibiotic, there has been extensive bacterial killing, although it did not reach also the basal line. When we're thinking why, we tried to find out the reason. You can guess why. Because the survivors all have the MDR plasmid. So the MDR plasmid made the bacteria resistant even to the effect of the nano-sized antibiotic. This took us to the, also we did a survival test on the rats infected with hypervirulent Klebsiella. The ones, 5 rats who took the conventional antibiotic, all of them died. The ones that, and the kidney, the lung was, as you can see, black because of the bacteria. But the ones who took the nano-sized, 2 of them survived. It was a 40% survival rate. It's not the best. It's not what we're looking for, but it's much better. And the lung looked like this. It was like healthy. So where does this take us? This takes us to our next step, our future work. We need to combat the circulating pathogen and the circulating plasmid. So what can we do? We have 2 lines. We will try both of them. The first one is we will use dual nano-sized antibiotics plus plasmid inhibitor, but we will compare it with the triple nano-sized antibiotic therapy, as all the community now are using triple antibiotic therapy for all the severe cases. I would like to thank my graduate students team, Dr. Safi, Dr. Hiba, Dr. Hadil, Dr. Rania, Dr. Riham, Dr. Shereen. All of them did great work so that we could have these marvelous results. Also, our collaborators from United Kingdom who brought us to the whole genome sequencing world and to see what's going on inside the bacteria, Dr. Radwa and Dr. Douglas, and thank you all.